There we go.
Hi, everybody. Welcome to the special webinar. We'll give people a couple more minutes to join and then we'll get started.
Oh, did I share? Okay. Cool.
All right, we have quite a few people, so let me get started. Thank you all for joining in. This is a special webinar hosted by the Society of Cryobiology and National Science Foundation ATP-Bio. So as many of you have heard, we made a bit of news back in April doing the first partial frozen kidney transplantation. I can claim no credit. The two people who can, McLean Taggart and Alvin Longchamp, will actually do the presentation. But before I introduce them, this was a first ever experiment in a porcine model. We are very excited that we are at this step. I want to emphasize that this is an early step. It's akin to Greg Fay's first vitrified rabbit kidney transplantations back a while now. There are certainly a lot of things that didn't go right, a lot of things we have to improve. This is not an unmitigated success and we're ready for clinical trial. There's certainly much more to be done. McLean and Alban are going to give you the details. This is the Society of Cryobiology, it's home. So we'll be open about everything that worked well and didn't work well. So to introduce the two speakers today, Alban Longchamp is one of our transplant surgeons. He was a fellow and he's joining us as an attending surgeon at the Mass General Hospital as of October 2025. And I can't be more excited that he's actually sticking with us. I may have bribed him with some beers on the way too. Alban is an excellent scientist. He has actually a great career in mitochondrial injury in kidney transplantation, effects of aging and gender, and a bunch of other things. This is also not the first time he was on New York Times. He was also on the cover because he was part of the team that did the first kidney xenotransplantation at Mass General, the first one and the second one. And we are very happy to have him here because I think he's in the middle of a transplant and may need to leave in a little bit. The other person, again, I need to give them credit. They deserve it, certainly, is McLean Taggart. McLean was actually a post-bac trainee with us. He has an NIH award for his studies too. He is joining PAN as an MD PhD student in just a couple of weeks. We are sorry to lose him, but we are also very happy he's going to be one of these special people that can achieve progress, and not just in medicine, but also in science all at the same time. So without further ado, gentlemen, the floor is yours. Thank you again for presenting to the society.
All right, thank you, Koke, for the introduction. So I'll start with, do you want to move the slide, McLean, since you have control? So transplantation is the best treatment for end-stage organ failure. I think with success come failures, and sadly, there's still more than 100 people in the United States only that are waiting for an organ. And consequently, 17 people die each day waiting for an organ. Next. And that's one of the reasons people are waiting so long for an organ. So in blue, you can see the number of people added to the wait list for kidney transplant, so in need of kidney transplant. And all of the other, so the orange and the green represent the number of transplants performed either from a living donor or from a deceased donor. And what's interesting is what you can see is that the increase in transplantation is far less than the increase in people added to the waiting list. Next slide. So we're trying to do better, and one solution to do that is to use more marginal organs. And you can see here in blue that the numbers of organs that are recovered that we're going for are also increasing in the past years. But the slope is much greater than the organs that are transplanted. So that means that we are going for more organs, but eventually discard these organs because of insufficient quality or inability to preserve them and maintain them in such a good quality that we consider them to be transplantable at the end of the transport, at the end of the storage period. And this is to show you something similar, but to show you all of the different organs and the evolution through the years. And what you can see is that 2 years ago, about 30% of kidneys are procured but discarded. Next. So as I mentioned before, one of the issue with a transplantation is that after procurement, these organs have to be transported to the recipient side. And historically, how that has been done is just to keep them on ice and what's called static cold storage. The issue with that is that induced during cold ischemia, a decrease in ATP level and alteration in mitochondrial health that subsequently during reperfusion at the time of the transplantation induced ROS generation, mitochondrial injury, and eventually cell death. Next slide. What you can see in this slide, and what's interesting for the kidney, is that the amount of cellular and organ injuries that during static cold storage depends on the amount of time the organ is stored. And you can see here the group minus less than 16 hours, 16 to 24, and increasing it up to 40. And you can see all of this endpoint delayed graft function or primary non-function, which is the graft that is never going to work, increase with increasing the amount of storage time in cold ischemia. And again, this leads with us trying to chase more marginal organs to increase discard and organ non-utilization. So one way to think about it is rather than to keep it on ice, is try to keep the organ alive outside of the body. And this is an illustration of the machine that was built by the Zurich group and Dr. Clavian's group that really is trying to mimic human physiology outside of the body. And you can see this is a very complex and expensive machine that includes a dialysis machine and virus infusion and monitoring capacity. And you can see that again with a lot of with a complex machine and a lot of testing and adjustment, they were in a proof of concept study able to preserve a liver alive, a human liver alive for up to 7 days. You can see here oxygen consumption that remains stable, the portal vein flow that remains stable, and histology as well as a tunnel that reflects apoptosis that showed preserved architecture and minimal apoptosis or cell death during these 7 days perfusion. So really a breakthrough study, but more so a proof of concept that comes with great cost and a complicated technology. Next. During much shorter time, machine perfusion and normothermic machine perfusion, specifically for the liver, has gained tremendous interest in the field of liver preservation. And there was 2 trials in Europe, and this trial that was conducted at MGH and led by Dr. Markman, that actually showed that in these marginal organs, in this older or organs from donor with some comorbidities, or DCD organs after cardiocirculatory death, that machine perfusion compared to static cold storage improved the quality of the graft and decreased complication. And again, this is a proof of concept from the Zurich group that if we kind of push the boundaries a little bit further, that after 3 days of preservation, they could transplant a liver with good function after transplantation. As you can see here, a decrease in transaminase and conserved synthetic function were shown here by a factor 5 and decreased in INR. The issue is machine perfusion can preserve the organ for a short period of time and restore energy, but ultimately, if we want to go beyond 24 hours or beyond a week, it's going to be complicated because these are very complex machine that requires a significant amount of people and adjustment every hour and constant monitoring. The other issue is transport, and this machine, for the most part, are difficult to transport and unstable. Next slide.
Okay, so I'll take over now. So we've established that there's quite a significant gap in transplantation regarding the number of organs that are available and the number of people that need organs. And we've shown that there are some active approaches to preservation through machine perfusion where you can keep organs alive for long periods of time. However, when it comes to thinking beyond scarcity, where we have enough organs for everybody, machine perfusion is a challenging solution because of how intensive it is. So if we try to think of alternatives to improving the preservation of organs, one of the obvious approaches is using a temperature-mediated inhibition of metabolism. Now, there's a rule of thumb that a lot of people in cryobiology, at least over here, like to say, and that's for every 10 degrees Celsius you decrease the temperature, your metabolic activity is about halved. So if you say 100% of metabolic activity at 37 Celsius, if you go down to the negatives, you're at 6, maybe 3%. And that's quite a significant jump. We keep the organs on ice around 4 degrees. So we have maybe 10% of metabolic activity. So if we can get a few-fold increase in that inhibition, we can significantly prolong how long these organs are stored for. Now, this is the thinking that our group initially thought of back in 2014 when we started looking into supercooling of whole organs. Now, supercooling is a very standard approach for a long time where you decrease the temperature below the freezing point, but you keep the system very stable. And what that does is it prevents any ice nucleation. So you're able to get the advantages of this metabolic suppression, but you don't have a lot of the ice-mediated injury that would occur if ice were to form. So back then, we developed this protocol where we load two separate CPAs, one of which is called 3-O-methyl-D-glucopyranose, or pretty much just glucose with a methyl group, and then PEG, which is just a long chain of polyethylene glycol. And we came up with this protocol where for about an hour, we load the organ using machine perfusion with this 3-OMG. And then we slowly decrease the temperature to right around -6 degrees Celsius, and the organ is stored with that polyethylene glycol. After the storage period of between 3 to 4 days, we bring it back to temperature, we do a recovery phase, and then we perform transplantation. Now, these two cryoprotectant additives were chosen based on some studies that some of our advisors and other groups have performed, showing that 3-OMG is able to enter glycolysis, but it's not able to be broken down. So what that does is it enters the cells into somewhat of a metabolic stasis, where the cells are trying to perform glycolysis, but they can't because they don't have glucose available. So it really inhibits metabolic activity and it enables prolonged preservation of cells. And polyethylene glycol is chosen because it's kind of like rebar for cells. It goes in and it stabilizes all the junctions and keeps the cells in place. So it kind of acts as this agent to keep the organs in their physical structure while they're being stored at such low temperatures, which can mess with membrane stability and transport. So it kind of just helps keep them alive. And you noticed I mentioned this recovery phase earlier. And now this is included because what we found is that after storage, these organs are in a very low energy state. They have pretty much no ATP left, and they're not able to perform very basic cellular functions. So if you were to go from -6 straight to 37 degrees Celsius, your mitochondria would have very high rates of reactive oxygen species generation, which would lead to a lot of cellular death. And what our group found about around 2014 and before is that if you perform perfusion after cold storage or supercooled storage at temperatures below 37 Celsius, so around 30 or 20, you're actually able to recover the energy stores. So you can build ATP while mitigating a lot of the warm ischemic reperfusion injury that would occur. So you don't get as much of that cell death, but you do enable the organs to recover their energy stores. And that just brings them to a better energetic state prior to transplantation. And you can see with reperfusion, if you have warm ischemia and SCS with no machine perfusion, you don't really have any survival. But if you add this 20 degrees Celsius, 30 degrees Celsius, we can get 100% survival out to a month following different storage times. So it's very important that after our storage phase, we include this to help get the organ ready for transplantation. So what this initial study showed back in 2014 is that using this -6 degrees Celsius storage period with the recovery phase, we're able to get 100% survival after 3 days of supercooling. Whereas with hypothermic preservation or SCS, you get maybe 50% after 2 days and 0% at 3 days. So this was a really groundbreaking study. It was published in Nature Medicine, and it really introduced the concept of cryopreservation in organ storage for transplantation to the world. It showed that static cold storage does not need to be the limit. You actually are able to push beyond 4C or 0C. And it really demonstrated that there's room for cryopreservation in the field of transplantation. And of course, after such astounding success, we were interested in scaling this process. We were thinking, okay, so rat livers are very small. They're maybe 10 grams in size. You can put them in the palm of your hand, but you can't put a rat liver in a human. That's not going to work. It's not going to be able to perform any functions. Are we able to scale up this process to the human liver? And that's what we showed back in 2019 when we performed supercooling of the human liver. Now, there were a couple modifications that we had to make to the protocol that I'm going to go over, but it follows the same general premise where you use a loading phase prior where you introduce 3-OMG, you perform the supercooling, and then you recover it afterwards with a subnormothermic perfusion. Now, one of the reasons we had to increase the temperature from -6 to -4 and add a few more cryoprotectants is because as systems scale, they get less stable in supercooled conditions. So whereas a tiny rat liver may be stable for days or weeks at -6, if you were to scale up the same exact system to the size of a liver, which can be, anywhere from 1.5 to 3 kilograms, so orders of magnitude larger, the systems get exponentially less stable. So in order to circumvent those problems, we added a few other cryoprotectants that are known to help both cellular health and system stability. So one of which is trehalose, which is an extracellular CPA. So it sits outside the cell and it helps to dehydrate the cells, and it helps during cryopreservation. It's a double sugar. And the other is glycerol, which is a liquid CPA. And that actually enters the cell and further dehydrates. And what we were able to show through this human liver study is that a lot of the key factors that you look at following storage were well preserved after the 24-hour storage period. Now, I should mention that 24 hours is a huge breakthrough because traditionally, when it comes to human liver storage, you want to transplant the organ under 12 hours. So anywhere from 9 to 12 is really the limit where surgeons are comfortable putting these organs back in the body. So the fact we were able to show that the energy charge, which is kind of like the ratio of ATP to ADP and AMP, is preserved after storage. We have the same bioproduction, the vascular resistance is preserved, the oxygen uptake. All of these are key indicators of how the organ is going to function. And we were able to show that after 24 hours of storage, which is almost twice, maybe 3 times as long as you could normally store these livers, we're functionally preserving the organs completely. So that demonstrated that, yes, you can supercool small organs, and with some slight modifications, you can also supercool these much larger systems that are more dynamic and challenging. Now, one of the problems when you think of long-term supercooling is that it is limited by stability. So whereas we were able to show these 24-hour storage periods, the systems get more tenuous as you prolong that storage period. So if you were to go out to, say, 3 days a week, you're not guaranteed that your system is going to stay in the supercooled state. You might have some ice nucleation. And that gets really tricky when it comes to thinking about real life usage of supercooled systems. Because if you procure an organ and you store it for this long period of time and you go to transplant it, but it's frozen, that's a catastrophic failure. And it really leads to a lot of difficulties when it comes to translating this type of technique into clinical practice, because you can't have someone prep for surgery and suddenly your organ's not usable. And also, you're limited in your thermal suppression because you can only go to around -4, -5 degrees Celsius. And as we mentioned earlier, that's really only maybe having the metabolic activity. If you want to bring it down to, say, 1% or less than 1%, you're going to need to suppress the temperature even further into the -10s, -20s. And that's really just not possible when it comes to supercooling. So our group started to think, how can we push beyond the limits of supercooling? And that's when one of the postdocs in Corkett's group, who's actually now a associate professor at Harvard Med School, Shannon Tessier, began looking to nature. And her during her PhD in their lab, they worked with this frog called Rana sylvatica, or it's the Canadian wood frog. And what it actually does is when it goes into hibernation, it will increase its intracellular glucose concentrations. So you can see in the leg, the heart, the liver, it just really fills the cells with glucose, and it'll actually go into a frozen state. So the entire animal is actually frozen, or what we call partially frozen. Now, the reason we call it that is because the theory behind the technique is that this increase in glucose, which is intracellular, will restrict a lot of the water into the endothelial space. So it's extracellular water. So what happens when the frog freezes is you're not having injurious intracellular ice formation, because that's the real culprit behind a lot of cell death. If you have ice forming inside your cells, you're going to have the membranes begin to lyse, and your system's just not going to survive. So what we believe the wood frog does, and the current hypothesis that we run with for our approach to deeper storage, is that by restricting water into the endothelial space outside of the cell, we're able to limit intracellular ice formation. Now, obviously, the frog has adapted its physiology to increase the intracellular glucose so high. You can't just replicate these systems in human cells because they don't have the function and the regulation to perform this type of uptake. So what we did is we developed a rat liver specific partial freezing protocol where we used all the lessons we learned in the supercooling development. We looked at how all the different CPAs functioned, and we came up with a custom cocktail that we believe is able to replicate a lot of the functions of this frog. So as I showed here, we have these permeating and non-permeating CPAs. And by using a combination of them, we're able to load the cells with these permeating CPAs and then load the extracellular space with the non-permeating. Sorry. And that will pull a lot of the water into this extracellular space, kind of replicating what you see with that wood frog. Now, this was a multi-year process. It took a lot of development. Dr. Tessier spent years working on it, and it's really a marvel she was able to get it working. And one of the key agents that they actually used to improve the outcomes is this component called SNOMAX, which is a bacterial protein. And what it does is it seeds ice formation at high temperatures. And the reason for that is because when you have ice formation, the temperature at which that nucleation begins actually changes the size of your ice crystals. And if you think about it, if you have ice forming in the extracellular space, you don't want these really long branches that are going to continue to grow and start to penetrate the cell membranes. So by using SNOMAX, we're actually able to minimize ice size, which can limit that that membrane penetration. And there was she performed a whole study where they looked at how SNOMAX affects these microvascular channels and showed that with SNOMAX at lower temperatures, you have much better survival in the endothelium specifically following storage. So that was one of the key components that helped enable this protocol to work. And they also had to do a lot of CPA optimization where they looked at different permeating cryoprotectants. So they looked at glycerol, ethylene glycol, and propylene glycol. And they are all different in their viscosity and how well you can perfuse them. Because one of the keys behind the protocol is loading the organ. You have to be very careful because when you have a lot of CPAs like you do in these systems, you're going to have very thick solutions. And you can't run thick solutions through an organ at high flow rates because you'll get a lot of shearing and sloughing of the endothelium. So through this optimization process, they found that ethylene glycol and propylene glycol were highly superior to glycerol because of that big difference in viscosity. You can see that after in the post-storage phase, the weight gain is significantly reduced with ethylene glycol and propylene glycol compared to glycerol. And other key organ functions like oxygen uptake, portal lactate, all showed that these two lower in viscosity solutions were better. So what they decided is based off the histology and all of these factors together, it appeared propylene glycol was the best permeating cryoprotectant. And then what they demonstrated is that compared to at 1 day storage at -10 degrees Celsius, -15 degrees Celsius had very similar functions. There was a slight increase in weight gain, but when it comes to a lot of the other indicators like lactate, oxygen uptake, and the actual appearance of histology, going to -15 was approximately equivalent to -10. And what that enabled them to do was actually push this storage period further back out to 5 days at -15 Celsius. And what they demonstrated is that the difference between 1 day and 5 days at -15 is almost negligible. So they were able to demonstrate that using this this modular partial freezing protocol, they're able to preserve the liver's health out to 5 days at -15 Celsius, which is really tremendous because that's longer than the supercooling period. And this is the first instance of organs being stored with any ice formation at all, because the rule of thumb has always been if you have ice formation during organ storage, then your system is not going to function. You're going to have to discard that organ. So this was really a groundbreaking study. And over the last couple of years, research fellow and I have been working to further improve this protocol and do some more translation. And we found that we can we can change the concentration of polyethylene glycol. And by doubling it, we were actually able to get better post-preservation outcomes. We had a lot of reduction in the weight gain that we showed was a little bit significant. And we also, for the first time, demonstrated that when you do a direct comparison of time-matched cold storage to partial freezing is very superior after 10 days of storage. You can see in pretty much every indicator of organ health, we have much better outcomes. The vascular resistance is significantly lowered. We have greatly improved oxygen uptake. Potassium is significantly lower. The transaminases, which are a cellular injury indicator, are significantly reduced. And that weight gain problem, it was 40% earlier at 5 days minus 15, is down to 0. So we were able to really improve a lot of these outcomes and demonstrate that partial freezing is vastly superior to static cold storage when it comes to these longer time periods. And then one of the questions we had is, if you think about it, we're by lowering the temperature, we're hoping to demonstrate that we're able to preserve the organ to kind of a pre-storage state. So we performed bulk RNA sequencing, and we looked at 3 days of storage. And if you compare partial freezing to SCS, and then these fresh organs, which are pretty much the healthy state of a liver directly out of the body, the partially frozen organs only had 117 differentially expressed genes, whereas static cold storage had 540. So that's a 5-fold difference in the number of differentially expressed genes. And what that shows is that following 3 days of partial freezing compared to static cold storage, we have a more well-preserved transcriptome. So the organ is in a state that is more similar to the pre-storage state compared to these statically cold stored organs. And that was really exciting. And just looking at some of the pathways that were differentially regulated, in partial freezing, everything is pretty much what you'd expect. We have a response to oxygen levels, so HIF-1 alpha, the traditional hypoxic response. Whereas with SCS, you have a lot of cell activation, immune regulation. So the organs are still functional, and there's probably a bit of this immune response starting to activate. So we were really excited about this because it is more evidence to show that using partial freezing, we're actually able to preserve these organs much better than static cold storage. Now, following this success, the next step is obvious. Can we scale these processes to larger systems? So can we do it with pig livers, human livers? And what we did is we took the rat system with the loading of polyethylene glycol and 3-OMG, the same storage period using 10% polyethylene glycol, 15% propylene glycol, Trailus and Snomax, and then that recovery phase following storage that I mentioned earlier. And we simply scaled the system to pig livers first, which are about 1 kilogram in size, and then human livers after, which, as I mentioned, can get up to 3 kilograms. And we were really excited because without any large system-specific optimization, we were actually able to demonstrate that this partially frozen protocol can be easily translated to these larger systems. So these are pig liver data following 10 days of storage, where we compared, again, SCS to partial freezing and then fresh organs. And you can see in all of those key metrics I mentioned, or oxygen uptake, partial freezing is better than SCS. Lactate, we see improvements. The energy charge is actually perfectly preserved compared to fresh organs. We have huge improvements in vascular resistance in both the artery and the portal vein. So the size of the vasculature doesn't seem to play too much of a role, at least for larger vessels. We have decreased weight gain. The bile production looks comparable, but when you look at the actual bile health, like bile pH, because you want to have alkaline pH, the bile being produced by these organs is significantly better than the statically cold stored organs. And even just looking at the histology, it's obvious, there's blood pretty much everywhere in the extracellular space in these static cold stored organs, but the partially frozen ones look quite comparable to our fresh organs. So this was really exciting for us because it means partial freezing is a very simple protocol to scale up to these larger systems, which is really exciting because that gives us hope that this simple protocol could be used for human translation. And of course, that begs the question, if we have success in pig livers, can we scale it up even a little bit larger to human livers? So here we have a picture of a partially frozen human liver, and this is a little video where we're kind of showing how you begin the thawing process. Now, you know, when you normally have ice form, ice is very thick and firm, and it's really difficult to break. But with this partially frozen ice, it's very light, it's gentle. It's a bit like a slushy or gelato. You can just pull it right off the organ. And again, upon reperfusion, we have very nice homogeneous perfusion throughout the organ. Um, sorry, it's very nice and red, which we're hoping for. We don't want any black spots of necrosis. And again, the perfusion parameters support that hypothesis. We have comparable uptake, oxygen uptake. We have similar lactate levels. The energy charge is slightly decreased, but not too greatly. The vascular resistance is very well preserved, flow rates, bioproduction, all of these key functions that we showed in pig livers are also well preserved between the pre and post freeze phases of these human livers as well. So this got us really excited. Now, obviously, when you think of cryopreservation, a lot of the early stages of developing these protocols is CPA optimization, because different systems and different cell types respond very differently to different cryoprotectants, just depending on where they come from. The bladder cells are going to respond differently to CPAs than the heart. So you have to perform these very extensive CPA optimization processes. And that's what our thoughts were throughout this process. We were like, okay, well, we demonstrated that partial freezing works well for the liver, but if we want to translate these protocols to other cell system or to other organ systems like the kidney, we're gonna have to re-optimize everything. But instead of just running with that assumption, we decided to be interesting to use some of our spare organs from the procurements that we were performing, just to see what happens if you were to try to do a horizontal translation to the kidney. So we perform the same general protocol of freezing and thawing. You can see the ice is very similar here. It just comes right off the organ with your fingers. We're pushing very gently here because we don't want to injure the organ by squeezing too hard. The ice just comes right off. And to our surprise, partial freezing actually appears to horizontally translate quite well. If you look at the actual organ perfusion, you can see with SCS, you have these very dark patches in 3 days and 10 days, whereas with partial freezing, you have much more homogeneous perfusion. It's much lighter. For 3 days, 10 days, even up to 30 days, you're able to get very homogeneous perfusion. If we look at these key indicators for the kidney, like again, resistance, you can see it's significantly reduced in partial freezing compared to SCS. We have slightly lower weight gain. The venous potassium, which is kind of a de facto cell health marker, is lower. Same with the lactate, so how ischemic these organs are, it's significantly lower. Creatinine clearance appears to be slightly better, and we have well-preserved urine production, whereas with SCS, you might have no urine. These partially frozen organs can produce urine even after 30 days. So this was extremely exciting because it means it's possible that for these different organ systems, you actually might be able to just directly translate the partial freezing protocol without doing cell type and organ type specific optimization. And even looking at histology, it's slightly decreased compared to SCS up to 10 days. And of course, having shown this for, sorry for these white boxes, I don't know where they came from. But having shown this for pig kidneys, we were interested, does this translate to human kidneys? Now there's one caveat for the human kidneys we get. When it comes to kidney allocation, traditionally, even kidneys can be used for quite a long period of time. We mentioned earlier 24 hours, but even up to 48 in some instances. So the kidneys that we get that are discarded for transplant are usually discarded for significant reasons. A lot of them have kidney disease, or the cold storage times prior to us receiving them are very long, way out beyond 36 hours. So the quality of these kidneys is quite low. But even so, performing partial freezing for 3 and 10 days, we see no significant differences in the resistance, in lactate, potassium. Urine production is preserved. There's a slight drop in creatinine clearance, but not a significant change. And if we look histologically, they look very well preserved even out to 10 days. So even with these injured, low quality human organs, we're able to perform partial freezing to quite a successful rate. So what that tells us is partial freezing is quite a successful protocol for these large systems, and it's very easy to translate. So, we were very excited with these results, and we were wondering, obviously, are we able to translate these organs? So we spent a few months, well, I guess a year, getting this pig kidney transplant set up. And we performed the first partially frozen pig kidney transplant. So you can see here's this transplanted kidney. It was stored just in a bag. It's partially frozen. You can see the whole prep. Right upon reperfusion, it actually pinked up, which surprised us because our first thoughts were this organ isn't even going to perfuse at all. It's just going to turn immediately black, and we're not going to get anything. But obviously that wasn't the result. There was, some perfusion, and unfortunately the organ didn't function as well as we would have liked it to for us to keep the experiment running. You can see after a while the organ did start to turn a little bit darker. But even as it was dark, we cut into it a little bit, and there was still quite good organ flow rates. So we were able to get, the organ was bleeding quite significantly when it's cut. It's not like the entire organ clotted up. So that's still an exciting result because it shows that these organs aren't simply failing drastically and clotting and being cut off from the system. And when we came back later and looked at the histology, we found a little bit of vascular congestion. So one of our current thoughts is it's possible that the microvascular endothelial cells are responding differently to the macrovascular cells, and we might need to do a little bit further protocol optimization for these smaller vessels to try to improve how they respond following storage. But regardless, this is a really exciting development for us because it's the first time a frozen organ has been transplanted, and it really marks a next step in organ cryopreservation and transplantation, where labs and our lab and other labs throughout the country are starting to think, how long can we store these organs and how can we transplant them? I mean, there was the paper that John Bischoff's lab published, I believe, last year, where they showed 100-day storage of the rat kidney with successful transplantation. And this is showing that cryopreservation is starting to become a serious field in transplantation. People are not just thinking, okay, how can these work in theory, but they're starting to think, how can we develop these systems that will actually work and can be used for our patients? So it's a really exciting time in the field of transplantation. And this leads us to a project that currently our lab is working on called New England Cryo-Xenotransplant. It's a grant that we're currently working on where we're taking a lot of these developments over the last decade or 2 decades, supercooling, partial freezing, and we're looking towards the also recent success in xenotransplantation, and we're thinking, how can we combine these into one field where we can really push beyond organ scarcity and enter a new era of transplantation where patients that need organs are able to get them as soon as they're diagnosed. And I'm going to hand it back to Alban so we can talk a little bit about the recent successes in xenotransplantation and how we envision the future of this field. You're muted, Alban.
One way to address this organ shortage crisis is to use alternative sources. So that's xenotransplantation, and more specifically from genetically modified pig. And our work has been mostly in collaboration with the Genesis, but that lacks key genes that are responsible for hyperacute rejection, in addition of other transgenes, human transgenes, that can mimic human organs for specific pathways like coagulation or inflammation, and etc. And as McLean mentioned, last year we performed the first kidney transplant in a living human, and you can see on the left the picture Dr. Kawai that led, and Dr. Riella, and as well as Dr. Elias and Mr. Sleeman, that really was a true hero in the endeavor. He passed away after 2 months after the transplant due to a sudden cardiac death that was mostly unrelated to the transplant. So, and then had before the transplant significant cardiovascular mortality. And it's really what this first case told us was that probably moving forward with xenotransplantation, we had to pick a healthier recipient, and that's how the team got approved for 3 additional patients to be performed in 2025. And so the second patient, Team Andrews, that you can see here, and that was really like a true historic moment when he threw the first pitch at a Red Sox game a few weeks back, and a whole month out of transplant. And just so you can imagine, before transplant, he was, extremely tired after dialysis, that he had to go 3 times a week, and could barely walk after dialysis, was in a wheelchair because he was just so tired and his quality of life was miserable. And so it really was, amazing to see him throw this first pitch, and after is now more than 150 days after transplant. So that makes him the longest human being surviving with a xenograft. Next slide, McLean. So I think the reason we're bringing xenotransplantation now, and we have this big plan for the future, is that the current model is that because of all the infrastructure required to produce these pigs and keep them in a clean environment, the model is going to be a few farms or one centralized farm in the US, and then the organs are going to have to be shipped in the US first, and then eventually worldwide. So the question of how to best preserve these organs is key. And obviously, if you're thinking even nationally, or more so if you go outside of the country, we're going to have to find solution to keep them to store these organs for at least more than 3 days. And that's when partial freezing or cryopreservation in general becomes extremely important. So that's really to illustrate what everything that has been said today, but current preservation technology are mostly limited to 24 hours, and comes with significant hurdle, including a large scale organ utilization and sharing. So really, with technique that can prolong that to several days to weeks, we can allow global sharing and the true banking eventually of organs, including xeno organs, including genetically modified organs for xenotransplantation. Thank you for listening, and I think we'll still have a little bit of time for questions.
Yes, thank you all for joining us and listening today. We appreciate it very much.
Thank you both for the talk. We'll open the floor to questions. If you type your questions in the chat, that's probably the easiest and most orderly way, given the number of people on the Zoom. But you can, if it's too, raise your hand, and I'll call the people with questions out.
So there's a question about vitrification. I think McLean touched upon it, at least mentioned the study. So yeah, I think it's very exciting. 100 days preservation is pretty close to, organ banking, and off-the-shelf availability of any organs for transplantation. I think it's still, not so much the freezing process, but the rewarming process is still challenging, and especially when we talk about, larger organs such as pig organs and human organs.
Any other questions from the audience?
So someone's asking
Yeah, so
about,
it's a good question. It's funny you mentioned allocation because Corkett and other team members have written a review that just got accepted on, not only allocation, I mean, not specifically allocation, but I think the ethical part of it. And yes, allocation will be completely changed because, it's based on distance, but the reason it's based on distance is because of our capacity to store organs. So if you can store it forever, I mean, it changes completely allocation. For example, for the kidney, the number of antigen mismatch you have is very important to your risk of rejection and long-term outcomes. So you could imagine a world where everyone get a perfect match kidney because you can just store it and wait for a perfect match kidney. So, and reduce immunosuppression, etc. And this number of antigen, goes into the allocation process. So yes.
I also want to touch on one thing regarding specifically to deceased donor organs. And it's an interesting fact we found is that the quality of the organ prior to storage is obviously indicative of how it's going to function after storage as well. So when we think about even allocating organs to different cryopreservation techniques, you're going to have to think about how healthy that organ is prior to storage regarding how you want to store it, because just because you're able to preserve function for so long doesn't mean that the function is good enough in the first place to be preserved. So it's going to be really important to think about what's the quality of this organ before we store it, and is it worth performing partial freezing or supercooling or whatever cryopreservation protocol you're going to apply before you actually apply it, because just because you can doesn't mean you should. So it's going to be interesting trying to come up with rules and guidelines regarding how you're applying these techniques and when you apply them.
I'll just add to that quickly before you move on to Zongu's question. Alex, thank you for the question. We actually have ambitions to design a global allocation system in the long term. We are working with the MIT Sloan School of Management to design that, especially for xenotransplants, to identify where and for which patients where maximum impact can be attained, what are the risks of variety of things, and what's the most ethical decision to decide. It's a little early on. We are just starting to talk about these things. The first paper is currently in like a very early draft. I'd be happy to share that when we have something a little bit more complete.
Yeah, and to get to Zhanghu's question, which is about the recovery of the organs prior to transplantation, we don't use any normothermic perfusion, but we do have a 3-hour sub-normothermic perfusion phase, similar to the protocol of supercooling.
Zonga, is that good enough?
Yes, that's perfect answer. Thank you. Is it blood contained? Does it doesn't contain blood?
No, it's
Acellular?
acellular.
Okay.
Yes.
I think for the kidney, using blood is a little bit more tricky than
Yeah.
the liver.
Yes.
Kidneys respond very weirdly to blood perfusions on the machine perfusion. I feel like livers are more consistent, and with kidneys, it's always a toss-up regarding how it's going to perform. So, yeah, that's definitely one of the annoying parts of working with kidneys compared to livers.
Yep.
All right, I think that's all the questions we have. Thank you everybody for being here and your time and your attention. If you have any further questions, just reach out by email and we'll be happy to answer any remaining questions as well. Thank you all.
Thank you, everyone.
Yeah.